Okay, let's make a start. Thank you for coming out on this very drizzly, miserable morning this morning. I hope you've enjoyed the last 4 lectures that we've had for the stem cell and tissue engineering block. From my perspective, we've had 4 incredibly inspiring leaders in the field of stem cells and also in tissue engineering, and I really hope you've enjoyed it. The purpose of this lecture today is to pull it all together, really, like Terry did for the metabolic syndrome workshop or metabolic syndrome block, where what I'm going to do is to revisit some of the concepts, but to frame them in a way that you can actually pull together those 4 lectures and integrate some of the new information from this lecture to be able to address the issues and applications of stem cells and your understanding in both the mid-semester test and the exam. And I'm going to give you more information on the mid-semester test in the next week, so don't be alarmed. So that's me. Email the subject email address, of course, if you have any questions. So here are my learning objectives. I'm hoping by the end of this block, and these are really learning objectives for this lecture, but also for the entire block. I hope that you'll have a good understanding of the key concepts in stem cell biology, and we'll cover some of those. That you will have gained an appreciation for tissue engineering, including its potential application, and design considerations and limitations as well. So it's not a cure-all, but you have to be aware of and be able to describe the limitations to the use of tissue engineering and use of stem cells as therapeutics, for example, and be able to discuss mechanisms or ways of overcoming those. I'd also hope that you'll be able to apply your understanding of stem cells and TE to all topics in frontiers and biomedicine and beyond. And so I'll give you a couple of examples towards the end of the lecture where we'll pull it together for a couple of examples of how it applies to some of the other blocks in this subject. So each of our lecturers have supplied you with learning objectives for their lectures, and I've included them in the slides here. I'm not going to go through them point by point, but the idea was to pull them all together into a central resource for you. So Melissa Little really talked to you about, I gave you a basic introduction to stem cells, the types of stem cells, their derivation, the differences between them. She talked about tissue-specific stem cells, IPS, didn't really cover somatic cell nuclear transfer, but also embryonic stem cells and the like. She also introduced you to concepts of organoids, which are self-assembling 3-dimensional structures that are being used, generated from both IPS cells and also from stem cells in the adult body, and how they can be used to screen for drugs and to look at disease model states, because you can isolate organoids from individuals from healthy tissues and also from diseased tissues. So she also talked to you about Waddington's landscape, and Waddington's landscape was what looked like the hills with the rivers through it, where she talked about the differences in potency, and she talked about reprogramming, and she talked about determination and differentiation as well. Megan Munsey continued on talking, but from a different aspect, which is ethical considerations of isolation and source and their application, and also the regulatory process. She also covered aspects of regulation, but also consumer expectations, media expectations, and from individuals who have illnesses where there is perceived benefit in stem cell therapies. So she really put into perspective some of the issues that Melissa covered in terms of where you isolate stem cells from and how you apply them and how you regulate them in a controlled way that's both safe and effective. Christine Wells talked about how she's using molecular profiling and molecular classification of stem cells to be able to gain more information from them. So one of the things that has sometimes been confusing in the stem cell field is to do with how you characterise and how you isolate your stem cells, and there's diversity in the techniques that are used and the source. And so she really talked about ways to be able to use the molecular profile of cells to be able to look for common features as a descriptive and predictive tool, really, through bioinformatics. So information such as origin, information such as function and equivalency, and also how to manage phenotypic diversity, but also to be able to characterise cells based on their molecular profile and be able to infer potential from that and the difficulty associated with those kind of decisions. Because we know that in order to define stem cells, it's not just about how they look, it's about how they behave functionally as well. So if you're trying to classify cells according to the molecular phenotype, you have to be able to be confident that there's robustness in those forms of assays to be able to predict function. So she talked about different platforms and initiatives that have been used, and these include Phantom, the Human Atlas, and also StemFormatics, which is the visualisation of molecular data about stem cells, and how that's empowering stem cell scientists to be able to, as I said, predict the function and to be able to classify the different cell types. And Christine also introduced you to this idea of having designer cells, which is it's not just about classifying the cells that we isolate, it's also about being able to find common transcriptional features of different cell types and then integrate those into being able to generate a cell of a new origin that doesn't, let's say, naturally occur in the body. And that might be integrating two different functions within a single cell. So really exciting developments in that front. And then yesterday, Andrea O'Connor talked to you about how the physical sciences and engineering are contributing to the advancement of medicine through their understanding of and research into materials and the composition of different materials and ways of being able to recapitulate them. And how there are different aspects of tissue engineering or considering what type of structures to use when you're trying to engineer a tissue from scratch, and how to overcome some of the hurdles that are faced in terms of trying to make a synthetic type of platform for cells in order to repair a damaged, injured or diseased tissue type. So I'm hoping already from those 4 lectures you could pull together the key aspects and be able to draw a narrative through them in different applications. So why is stem cell research important? It's important, it's gained lots of exposure over the last 15, 20, 30 years. But why, what sort of information? It's not just, does it give us, it doesn't just give us information about therapeutics. It's far more broad than that. And that's because stem cell research helps us to understand basic biology, such as cell fate decisions, what is involved in determining a cell fate from one particular cell type to another. How does development work? How do tissues form? How do organs form? What's the arrangement, the organisation of these processes? Also, regular homeostasis. So how do tissues maintain their integrity and how do they turn over? How do they respond to stress or to trauma? So it's maintaining the steady state but also responding to trauma, injury or a requirement for replacement of tissues. So it tells us about all of these key decision-making processes in both human development and maintenance. And so if we understand how these systems work or these processes work under normal circumstances, we can then have a comparison for when we're trying to understand disease states. And this is really advancing, as I said, with the use of iPS cells, with organoids, particularly because you're able to directly compare not just single cells or a monolayer of cells in a dish. You can actually see how, for example, epithelial cells from all different organs function in a normal state and in a disease state. So you start to recapitulate what's going on in vivo. And that's very useful when you're wanting to understand disease and to develop cures for them. You know this, you've heard this a million times. What is a stem cell? We have these broad categories through which we define a stem cell, and that is their ability to self-renew, make more copies of themselves, to be able to generate a large number of progenitor cell types which have the capacity to differentiate into multiple different cell types. But the key to this is really about how they function, how these cells function and what they do. And we need to demonstrate that in vivo. So it's really, stem cells are operationally defined by their function, how they respond, how they produce mature, lifelong cells, and how they respond to injury. And you have to be able to demonstrate that to demonstrate a stem cell. Melissa covered the classical stem cell properties other than self-renewal and being able to produce multiple different cell types. She talked about these cells being able to proliferate extensively, that they can clonally repopulate. That means you can take a single stem cell, you can culture it, you can then take those progenitor cells and you can reculture them, reculture the single cells, and they'll form more colonies. Okay, so clonal repopulation. Or where you can put a single cell and it can repopulate a whole tissue, like we use for hematopoietic stem cells to determine their function, is that you can put a single hematopoietic stem cell in an irradiated mouse and it will reconstitute a completely ablated bone marrow. They're capable of multi-lineage differentiation. Present in low numbers, not always. Think of the gut. Quiescent, not always. Sometimes they're actively proliferating because they're having to maintain and turn over a huge amount of cells. They reside in a niche. So this is a specified microenvironment, which I'll talk about in a moment. They tend to be morphologically unrecognisable, so they don't have identifying features about them and lack phenotypic markers, which has been one of the difficulties in being able to isolate these cells. So classically, and this is an example of an adult tissue hierarchy, adult stem cell hierarchy, they sit in this hierarchy of, you might have different types of undifferentiated stem cells living within their niche, and they can respond to proliferative cues within the microenvironment to produce progeny, which may start to become determined down a particular lineage. And these can then produce multiple different cell types. So you were also introduced to these, so from these intermediate progenitors to these fully differentiated, mature functional cells, but that it wasn't a unidirectional process, that there could be both transdifferentiation, where one cell type transdifferentiates into another cell type in response to stress. So, for example, in the lung, the type 2 pneumocyte can produce type 1 pneumocytes. But also you can have dedifferentiation. So if you take particular cells, I'm thinking of chondrocytes, they'll dedifferentiate to a less specialised cell type if you put them in culture. So they become less specialised. And then, of course, the ultimate there is where for IPS cells, where we've taken a mature cell type and reprogrammed it back to an undifferentiated cell with multiple, or that is pluripotent. So as you get restricted, so lineage restricted down a particular pathway, determined and then differentiated, you lose proliferative potential. So there's that balance there between specialisation and ability to proliferate. There are different types of stem cells, isolated from different sources, that have different properties. They have different advantages and disadvantages. And if you think of the hierarchy of potential, we introduce you to totipotency, pluripotency, multipotency. So think of a totipotency being an example of that being a fertilised egg. So totipotency means ability to be able to produce all of the cell types and extra embryonic tissues. Pluripotent stem cells, an example of this are ES and IPS cells that are able to form the 3 germ layers, so can produce all of the cells of the organism, but they can't give rise to a fully functional individual, and that's the difference between a totipotent stem cell and a multipotent. Totipotent stem cell, if you put it in a suitable maternal environment, will produce a new individual. And then we have multipotent stem cells. An example of that is a mesenchymal stromal cell, which has the ability to produce lots of different cell types, but is restricted in its ability. And the different types, tissue stem cells, embryonic stem cells, IPS, and you should know how those are derived and the differences between them. So what determines stem cell behaviour? I talked about, and others have talked about, the microenvironment or the niche. Stem cells reside in a niche. It's a microenvironment consisting of multiple different factors. They're both cellular and acellular factors. They interact with, so within the niche, cells interact with other cells. They interact with extracellular matrix molecules, which is a mesh-like network that produces the structure and an acellular part of a tissue. They interact with soluble factors and also sequestered factors that can be within the matrix. And it's actually the niche that controls how the stem cells respond. And this environment can be both restrictive and permissive. So by that I mean, under steady state normal conditions, let's say a hematopoietic stem cell will reside within the endosteal niche in the bone marrow, and that environment is called restrictive. So it's restricting it because it's saying, stay quiescent, stay quiet, we don't need you at the moment. And then in response to soluble cues and cellular cues, those cells can be activated, so that environment has changed to a permissive environment where that stem cell can respond, proliferate, produce progenitor cells, and reconstitute cells that are required that have been depleted in the bone marrow and blood. And this sort of management control is highly regulated. It's very complex. The extracellular matrix is a network of macromolecules. Consists of, for example, with cartilage, you have lots of water, you've got collagen fibres, glycosaminoglycans, and you've got this meshwork. And you often have cells because a tissue, as you know, is not made up of just cells. So the distribution and composition of the extracellular matrix is unique and different in different tissues, and it can consist of different ratios of these type of extracellular matrix molecules, which is why it's really important when you're considering this for tissue engineering that you choose the appropriate ones. And this is a key point. Seemingly equal cells can behave differently depending on the microenvironment in which they're placed. And this is one of the challenges early on with stem cell therapies, is that people were putting cells into an environment where there would be a heavy inflammatory response, and the cells would just respond in, for example, a fibrotic way in response to the environment in which they'd been placed. So this concept of the environment controlling cell fate decisions is a key one, and it's a key one when you're looking to use stem cells as therapeutics. And these cells will respond to not only the biochemical components, but also biomechanical components of that tissue type. Don't expect you to remember this. What determines cell fate? How do we grow and control stem cells in the lab? Well, we can get information from a range of sources. We can get information on phenotype from using flow cytometry, which is where we use cell surface molecules to be able to identify particular cell types. We can separate cells based on their expression of cell surface molecules. We can look at expression studies to determine gene regulation, and we can also determine potential through in vitro functional assays. So it's actually, we need a combination of in vitro assays, in vivo assays, and both molecular and genetic profiling to be able to pull all that information together, to be able to form these complex hierarchies. So we don't always have to do this from scratch, and you'll learn that, and you will know that when you're looking to address a problem, you have a look at what's around in the literature, or you have a look at what's known already. So when we're, as stem cell biologists, trying to determine these hierarchies, these pathways, we actually can take a lot of cues from morphogenesis, so from development. And that's because repair recapitulates ontogeny, which is repair processes often demonstrate the same molecular processes and cell fate decisions and differentiation processes as we see during development. And so that gives us an idea of from studying development what sort of cues are involved, whether they be cellular cues, interaction with matrix molecules, secreted soluble factors which control the proliferation and the differentiation. And this is an example of branching morphogenesis in the lung, so the formation of the tree-like structure in the lung during development. And we know that there are key interactions between epithelial and mesenchymal stem cells at the bud here during the elongation process, but also during the differentiation of these progenitor cell types into mature mesenchyme and mature epithelial cells. So that's how we get a lot of our information. So what do we do when tissues or organs fail? As you know, we live in an ageing population. We have an increasing population, which I'll cover at the end. And one of the consequences of living longer is that our tissues and organs and their function can fail. So at the moment, whether it's due to lifestyle factors, genetic factors, injury or trauma, the ways of replacing tissues or organs usually include either transplantation or the generation of a prosthesis. So with transplantation, of course, it requires a human donor. And there's a paucity of organ donors. There's the risk of organ rejection because you're using an organ from another individual that would be recognised as foreign. And you can ultimately have transplant rejection where it actually causes harm to the individual through a tissue rejection process. So the other option is to generate prostheses. And so they're often a replacement, and they provide often structural support, but they don't often they're not a biological replacement, and they also don't necessarily give you full function. So there's some examples here of a prosthetic knee joint, and we've got a heart valve here, and we can make prostheses for all sorts of critters. And tissue engineering stepped out of the light and in the 1990s with Vacanti and their paper in Science, which was really paving the way for this new field called tissue engineering, which was going to use a biological approach to be able to repair. And so it could maintain, repair, improve, or replace damaged or diseased tissues or organs. You might have heard of this term regenerative medicine, and regenerative medicine is a more specific form of tissue engineering, which is the use of stem cells in tissue engineering. Okay? So if we have a look into the Bible, and here's a lovely plate of a description in the Bible, one of the first mentions of tissue engineering really happened here in Genesis. And this was where God came, and there was Adam, and he took part of Adam's rib and he made Eve. And in my mind, that's the first evidence of tissue engineering. We know a little bit more about biology now. You might question whether that would be achievable, but it is the first reference that I've found. And so many thousands of years later, here we are trying to engineer tissues, and we've been largely unsuccessful. So there have been some successful studies in tissue engineering with clinical applications, and here are some examples. So there's been some success in generating skin or transplants for skin. These include epicell. So epicell contains autologous keratinocytes, which are grown in a sheet and then placed over the surface of damaged skin or disrupted skin that's unable to heal. The dermal regeneration template doesn't contain cells, it's just a matrix. So it has a silicon membrane, which is a protective membrane, but there is a cross-linking network here of collagen and glycosaminoglycan from sharks and from cow. And then there's appligraph, and appligraph includes both cells and also matrix molecules. So it's a collagen matrix with dermal fibroblasts on it. And they've been relatively successful in being able to help repair skin where it's unable to, because if you damage skin beyond a certain surface area, it's unable to regenerate. So the second tissue that was reported to have some success was cartilage. So my PhD was actually dedicated to trying to develop ways to tissue engineer cartilage, and it's quite tricky. The studies use chondrocytes, and I was trying to use mesenchymal stromal cells, but there's been some success. So Carticell uses autologous cartilage cells, and you can think how precious cartilage is, and isolating those cells can be, you have to harvest those cells in a very careful way to not cause any further damage. And then it's integrated with some hyaluronan, which is a naturally occurring extracellular matrix molecule found in cartilage, and this has had some success in repairing articular cartilage surfaces. And they've been successful, well, I'll come back to that. They've been successful really because they're thin and they're flat. And if we think about, okay, so we've got, let's say, a retinal surface, or we've got a thin cartilage surface or skin. If we think about how we're going to scale that up to more complex structures, you can see that we require increasing functional parameters, more metabolic requirements. There are more different cell types involved, and you're going to need to start considering inter-organ interactions. So you need further complexity if you go from a flat structure to a hollow structure. So a trachea would require a couple of different layers. If you're thinking about a bladder, then you would need some inner epithelial cells. You need smooth muscle around the outside. It would need to have functional capacity. It would need to be able to hold liquid or hold urine and respond to different cues to be able to either contract or relax that smooth muscle as appropriate. And then we get into even more complex solid organs such as the kidney. And this requires interaction of a huge number of cells with precise control, precise architecture, and both cellular arrangement and three-dimensional structure. So it starts to become much more complicated. And one of the main limitations is this absence of vasculature or difficulty in producing vasculature. You need vasculature to provide oxygen, nutrients, to clear waste products. And you can only get diffusion of oxygen and nutrients and clearance from very, very thin structures. So you do need to have a vascular supply. So ways that researchers are trying to overcome this lack of vascularisation, because as Andrea mentioned, originally it was just you threw some cells with a scaffold and you put them in and you wanted to wait until there was an endogenous blood supply produced. And that took a long time, and by that time those cells were hypoxic and died. So some of the approaches have been to use soluble factors such as VEGF to stimulate angiogenesis within the tissue, or to place the tissue-engineered construct into for example, a bioreactor, which would mimic the in vivo environment to help trigger formation of new blood vessels and to generate a blood supply. But the most successful to date really has been the transplantation of tissue constructs into living organisms, both animals and into humans, to generate an autologous endogenous blood supply prior to transplantation to the site where it's required. So this is an example here of, this was published in 2012 from Johns Hopkins, and what they did is they tissue engineered an ear, and then what they did is they transplanted the ear into the recipient's arm, and they waited for an endogenous blood supply to be developed in the arm, and then they took that ear, they did a skin transplant over it, and they actually through surgery, attached that to a woman who'd had a terrible accident and lost her ear. So that has been the most successful way to produce a blood supply, and that was a really revolutionary approach. So one of the things we also know is that cells can respond to different mechanical stimuli, and this is where the use of bioreactors has come into play. So we know that different cells require different, if you're going to grow them in vitro, that they require biophysical or mechanical cues to maintain their differentiated state or to encourage them to differentiate into the required tissue type. These can include cartilage requires hydrostatic pressure or compression. We know that muscle requires cell stretching in order for the alignment of the muscle fibres and to maintain those myocytes as differentiated cells. We know that if you're thinking about blood vessels, you've got to consider factors such as unidirectional flow past cell types. So we need to mimic the environment in which those cells are in vivo in an in vitro setting. So what I'd like to do now is to spend a little bit of time. I'm not going to talk about the basics of tissue engineering from a traditional approach. I'm going to get you to think about the different components and how they're required and try and pull together a little bit of stuff in regards to considerations that Megan brought up about isolation, source, ethics, etc. and arrange. So this was the traditional tissue engineering approach. You took some cells, you added some soluble factors, and you put them in a scaffold and transplanted them. And we knew that there was some importance of biophysical properties, so we knew that the scaffold had to be similar to the tissue type you were transplanting it into, and you could, using some bioreactors, introduce some biophysical properties before you introduce the cell in vivo. And this seems straightforward. Okay, I've got to consider 4 different things. It's actually so much more complex than that. So what's the first consideration? First consideration is, what type of cells am I going to use and I need? Where am I going to get them from? What are the considerations in a therapeutic application for the choice of cells? So you need to collectively, we need to consider whether we're going to use autologous cells, that are cells from the individual's own body, or whether to use cells from an individual of the same species, which is called allogeneic cells. This has really been, the use of autologous cells was revolutionised by Yamanaka and Thompson publishing their papers on iPS cells, because for the first time it meant that you could actually derive cells that were not even multipotential, pluripotent in their potential, as a possible therapeutic application. So before that, the focus was on a lot of adult tissue stem cells, which had fairly limited regenerative capacity or proliferative capacity, but now this has opened the door for use of autologous cells that can be derived from reprogrammed somatic cells from the individual. The other option is the use of allogeneic cells. So this means that you could have banked stem cells. So you could, for example, generate a whole bank of, let's say, osteocytes or osteoblasts that you want to use in times of crisis where you don't have time to be able to proliferate in culture the required number of cells. So they can be from you or from another individual, and then you'd need to have a think about whether you would use undifferentiated cells. So would you use a stem cell like a mesenchymal stromal cell, an iPS cell, an embryonic stem cell, for example, or pluripotent cells derived from somatic cell nuclear transfer? Would you use them directly, or would you need to differentiate them before you transplant them? There are issues with safety, obviously, if you're going to transplant undifferentiated stem cells into an individual, because they will respond to the microenvironment in which you place them. So that's got to be a major consideration. Or do you use differentiated cells, terminally differentiated mature cells that can be derived from your undifferentiated cell types? And you would need to consider where you would isolate them from. If you're going to use autologous cells, you want to make it a non-invasive procedure. Where from, what part of the body, and this is more specific to adult stem cells or tissue stem cells. How do you enrich? So you need to isolate them. You need to purify them. you need to enrich them. And that means by enriching, it's that you're actually generating a relatively homogeneous population of cells. How do we generate enough for transplantation? If you're taking a small biopsy and trying to expand cells, how long is that going to take? Is that going to take you 6 months? And is that going to be appropriate for the condition you're trying to manage? How do we guide the cells to differentiate and maintain the desired phenotype? How do we make sure that if we're going to take a pluripotent iPS cell, that we differentiate it to become an islet cell and it maintains that phenotype? Not that when we put it into the body, it's going to de-differentiate, which is possible, or cause damage or proliferate uncontrollably. So how do we manage that? How do we deliver them to the correct location so they don't just get cleared by the body or recognised as foreign? And how do we make sure that they can survive, mature and function as well? So this can take a huge amount of work and a huge amount of considerations in how to do it. So here's a quick example. If you need the cell, do you need the cells immediately? If yes, it's going to make a difference whether you need a lot of cells or it's a large organ or a small organ, as to whether you use adult stem cells, banked iPS cells. Obviously, you could bank iPS cells in advance and then differentiate a large cell number. If it's a small structure, you could use autologous adult, so from the individual, or you could use allogeneic. And if you don't need them immediately, then what type of cells do you choose if you have the time to be able to expand them? You'd also need to consider whether you need to incorporate signalling molecules to drive differentiation or to maintain a differentiated state. And these are often, there's some usual suspects, which are the bone morphogenic proteins, FGF, which Andrea talked about integrating into some of her scaffolds, VEGF to promote angiogenesis, and TGF-beta. And these are often, as I said, information is gathered from them based on their role in ontogeny and morphogenesis. And you can integrate or incorporate these molecules into your scaffold fabrication as well. I won't cover this because it was covered yesterday, but this is just to help your revision as well. So I know that these slides have a lot of information on them, but they are to help your revision process. They need to, the choice of scaffold needs to reflect the application. And they can be natural or synthetic, and there's great sophistication now in being able to design or manufacture different scaffolds with the desired properties that can be changed with very subtle alterations. So one of the, an aspect that's gained a lot of interest of late is the use of decellularized tissues. So we know that there's conservation between different species in the constitution of the extracellular matrix. The idea of being able to generate a whole organ from scratch is mind-blowingly complex. And so one of the areas of interest has been to take a tissue or take an organ and then exposing that organ to a range of processes to remove the cells. So to remove all the epithelial cells, all the mesenchymal cells, through a process of temperature, treating with detergents, use of nucleases. There's a few different techniques. And what it basically does is it strips all of the cellular content out of that organ. And what it leaves you with is this perfect structural, extracellular structural matrix, which resembles the original organ. And the idea is that then you could introduce, so this acellular scaffold, you can then introduce the recipient's cells, you can reconstitute the cellular compartment or the cellular compartments of that organ, and then transplant it back in. And so because you're retaining the extracellular matrix, you're also maintaining these mechanical properties. And here's an example of both lung, heart, liver and kidney, which have been decellularized. So they still have the structure of blood vessels, they still have the structure of the mesenchyme, but through the matrix, but there are no cells in there. And so the topology is maintained of this organ. As I said, it's highly conserved, but there's no endothelial cells. So one of the limitations has been when you transplant, when you repopulate with cells and transplant or culture, that you get thrombosis because you don't have those endothelial cell surfaces. And this approach has been successful for a few minutes to hours. So that's it at the moment. And that is because of issues such as thrombosis. You can, if you're thinking, if you've got your ethical hat on, you might consider, all right, well, if we need an organ to start off with, where does that come from? And so you might think, so what's the source of this decellularized organ? Does it come from an animal, for example? Does it come from organ donations? And other options as well. Andrea talked a bit about bioprinting. So she talked about how you can print three-dimensional structures and you can control gradients of cells, gradients of soluble molecules, for example. And what you can actually do is precisely control the architecture of a structure. So this is a hydrogel, and you can see here that we've had cells which have been deposited. These are smooth muscle cells and fibroblasts. And this paper actually shows that you can either have alternating regions of fibroblasts and smooth muscle cells, or you can have, for example, an inner layer of smooth muscle cells and an outer layer of fibroblasts. So you can get quite sophisticated constructs through this three-dimensional printing process. So where have we been? Well, we've just finished the stem cell block, but before that we had the metabolic syndrome block. Does that mean that they're discrete? No, they're not. You've learned about the metabolic syndrome and the 4 risk factors or contributing factors to the development of metabolic syndrome, or the classification of it, including obesity, insulin resistance, dyslipidemia and hypertension. And you know that these different conditions can result in a range of pathologies, which can include things like diabetic nephropathy, retinopathy, neuropathy, skin ulcers, etc. So if you're considering stem cells, you might think that there would be potential to use stem cells as a therapeutic for metabolic syndrome associated conditions related to diabetes and cardiovascular disease. And so if you go to the clinicaltrials.gov or have a look at Stem Cells Australia's listing of clinical trials, you'll see that actually diabetic neuropathy, nephropathy, retinopathy, vascular disease, ischemic heart disease and stroke are all there are clinical trials using stem cells at the moment. Cynata was mentioned by Melissa, and this is where iPS cells are being differentiated into mesenchymal stromal cells, and these are being used for their immunomodulatory properties in graft versus host disease. So here's a really exciting development that relates to metabolic syndrome. This product, ViaCyte, is just entering phase 1, phase 2 clinical trials. The way it works is that they've taken human embryonic stem cells and they've differentiated them into pancreatic progenitor cells and to mature islet cells. They then take these cells and they've implanted them within this permeable membrane, which is called the encapture device. And then they take these cells within this device, within this capsule, and implant it within an individual with diabetes. What happens is that this capsule can actually allow different cellular components to be able to diffuse across this membrane. And these differentiated islet cells then can respond to, for example, the blood glucose level and start to produce insulin, which can then permeate out of the membrane into the individual's blood system. So it's a really exciting development. You might be thinking, well, you would probably have, it would be recognised as being a foreign foreign object, and especially your cells would recognise the allogeneic embryonic stem cells. Well, that's why they're kept within this device. So the immune cells can't permeate the membrane and therefore destroy these islet cells. It's really exciting development. So obviously another option is to engineer new tissues for transplantation. I'm going to talk about respiratory stem cells when we get to the respiratory health block, and how there are potential biological applications for stem cells in the management of asthma, COPD, and for transplantation. I'm going to spend the last 5 minutes, bear with me, on a slightly left of field approach to tissue engineering. So our population is currently at about 7.5 billion. When you were born, it was about 5.8 billion. And it's set to reach 9 billion by 2040, which is twice the number of people from when I was born. And so one of the real challenges is going to be to provide enough food for 9 billion people and more. So with advances in stem cell research, there's been a real drive to the potential application of tissue engineering practices to make a food source. And that is the tissue engineering of animal-derived protein. So this push has been for a lot of reasons. So we know that a third of the land surface is used for the farming of animals for consumption. We know that the amount of water, so for a 110-gram burger, it requires 3 kilos of feed, 200 litres of water, 7 square metres of land, and produces 6 kilograms of carbon dioxide into the environment. So there's a real push from environmental concerns, and there's also concerns about the slaughtering of a sentient being, for example. So there's a real drive for this. You can have a look at this in your own time. If I were to eat a 110-gram portion of beef, it would be equivalent to me driving to work in equivalent carbon dioxide emissions. So the process of tissue engineering meat really comes from taking a tissue biopsy from whatever animal it was that you wanted to make protein from. Take a small biopsy and you isolate the microsatellite cells from that. They're highly proliferating progenitor cells from, that can produce myocytes. So you take out the myocyte cells, you proliferate them in culture and add growth medium to them. And then what you do is you make lots of them, and then they start to assemble into myotubules spontaneously. Then you take those myotubules, you place them on a 3-dimensional matrix. You subject them to, for example, cooling and heating, which induces a stretching or relaxation contraction process. They start to form muscle fibres. You take a whole cluster of those muscle fibres, add some vitamins, mix them up, and then you've got a sausage or a burger, for example. And the first demonstration of this was in 2013. So Mark Post in the Netherlands actually took myosatellite cells from a cow, and he generated 20,000 muscle strips. He put these muscle cells under tension, and they differentiated into muscle cells. And then he took 200,000 of them, mixed them together, and they filmed this burger being cooked and eaten. The cost then was $325,000 to make that burger. 2 years later, it would cost $10, and that's the advancement of technology in this area. There have been other approaches. Gabor Forjak had a tissue engineering company called Organovo. He started a company called Modern Meadow, where he actually 3D printed some muscle, and he cooked and ate it in a TED talk where he was talking about tissue engineering, and here's the link. They've actually moved away from food manufacturing to printing leather now, so bioprinting leather surfaces. You may have read on the ABC and also in The Age this week about this company called Memphis Meats. So Memphis Meats are a startup company that's just been, they've raised $21 million Australian for their startup company. Their two key head people are a clinician and a stem cell biologist. And they've raised this money from Sir Richard Branson and Bill Gates have given a large component of this money, but also a company, major meat company in the United States. And so they've actually demonstrated that they can tissue engineer beef, chicken and duck. And they've got recipes there and show how they prepare it. It actually looks pretty good. And then Mark Post's company, Moser Meat, will be launching their website very shortly. So I hope that's given you some insight onto actually how we can use stem cell, our understanding of stem cell biology and tissue engineering for purposes far beyond just repairing humans. We can actually feed populations as well. So these are the summary of these 2 lectures. If you're interested in research into any of these areas from this block, follow this link into stem cell projects for honours and masters. Thanks everyone.